my group, we're in the physics department at UCSB, and we do a lot of broad interdisciplinary work. And faced with a half an hour or 35 minutes to talk to you about robustness trade-offs and tipping points, I chose to talk today most specifically about how robustness trade-offs play out and can lead to tipping points at some level in adaptive immunity. So I'll mention a little bit some of the other kinds of things that we work on, and also give all the credit to my two former student and current student Sean Stromberg and Kimberly Schlesinger, who were instrumental in all of this work as well as the artwork. Okay, so in my group, we've been thinking a lot about dynamics and robustness, fragility trade-offs, and multi-scale modeling, and how these concepts play out in a broad set of different kinds of problems. So earthquake dynamics is something that we've examined from sort of the granular scale all the way up to the scale of dynamic fracture on earthquake faults and hazard analysis. We've also looked at forest fire models, models of fire propagating, statistics over time, disturbance ecology as a way to think about how ecosystems evolve. We've looked a lot at biological networks. So today I'm going to talk to you about some of the work we've done on the immune system, which is an example of kind of forward modeling that we've looked at in this area. A lot of our recent work has been on neuroscience. So this is another one. This is a really data-driven field for us, at least at the moment, where we're developing methods for dynamic networks to try to understand sort of switching behavior in the brain between tasks, as well as relationships between structure and function. And then finally, we work a lot on technological networks, the internet, communication networks, and also look at group decision-making and the influence of social information in collective decision-making and problems involving risk and threat, like evacuation, for example. Okay, so a common theme, there's sort of two common themes within our group. One is this idea of trying to look at problems from one scale to the other, and then the other is to really think about how the trade-offs at each scale can lead to structured responses and to some sort of environmental driving. So this is really an idea that I've been working on for a long time, for, I don't know, at least 15, 16 years. A collaborator of mine is John Doyle at Caltech. And we came at this problem from two separate sides. One from the sort of the physics side of developing a sort of simple mechanistic understanding of complex processes. And the other side really was this more engineering side, the side that's motivated by complexity as it arises in technologies or in biology, where there's an enormous amount of hidden structure in many cases that give rise to responses, high variability response, in part because you're embedded in a highly variable environment. So the idea of this hot mechanism that was mentioned before is that complex systems in biology and ecology are really driven by this kind of complexity, not just a phase transition in a gas, but rather something that's evolved structure over time to a state that is tolerant to uncertainties in the environment and components, and yet has fragilities that are also associated in many cases with these sort of tolerance mechanisms. And that this leads to specialized structure, which often with a great deal of hidden complexity that may give rise to new sensitivities or unknown or neglected perturbations and design faults. So this is one of the reasons you really have to care about complexity when you do something like bioengineering of the ecosystems, is because they are so complex. And so you go in, and as was mentioned in the previous talk, this is a really important thing to understand these structured complexity in your system. So they're robust to the kinds of complexity that are common, the system was designed for or is evolved to handle, yet fragile to unknown complexity, rare events, things that have changed on the time scale of the evolution of the organism. And my perspective is really that this is the most important feature of complex systems and the essence of hot. And this cartoon here is actually kind of planted because it shows a fragility of something like wildfires, natural terrestrial ecosystems, which are subject to disturbance, have evolved certain tolerances and plant types in response to their natural ecosystem, which is subject to disturbance like lightning and fires. And as soon as humans go in and intervene in these ecosystems, there's the possibility of new fragilities that, and type conversion and things that we may not have anticipated. Okay, so what I'm going to focus on today, though, is really the immune system. And so this is another area where we've been thinking about this hot mechanism, thinking about dynamics and modeling. And really, it's a natural for thinking about how this kind of evolved complexity, because the adaptive immune system in an organism is something that on the lifetime of the organism, it adapts to develop memory cells, to previous exposures, pathogens that it's seen in the lifetime. And so it's a natural mechanism for this kind of evolution of robustness and fragility. Okay, so the idea is that we're going to look at basically various phenomena. I'm going to just focus actually on two today, of basically a model of the immune system, the adaptive immune system, which is based on understanding of the mechanics of different cell types. So it's a population dynamics kind of model. And the idea is we're going to use this model to understand how the immune system solves problems, predict failures that could result from the natural problem-solving mechanisms themselves, and gain insight both into things that would be useful in terms of understanding the immune system, therapies, diagnoses, but also broader implications for complex systems where you have adaptation and co-adaptation taking place real time. Okay, so four of the areas that we've thought about, the first one and the last one are the ones I'm going to talk about today. One involves the sort of adaptation over long time scales, so say the lifetime of an organism when it's exposed to a variety of different infections. The second one here has to do with zooming in on the idea that you may have one exposure and then you have another exposure soon after that. So this is relevant for things like immunizations to the flu this year, but also to long-lived chronic viruses, say like the dengue virus, where there's different epitopes of a similar infection. And it turns out in this particular case, you can have protection, it can be a neutral secondary infection, or you can actually develop a more severe reaction based on the prior reaction because of the holes that it causes in the immune system. Another thing that we looked at is this notion of the hygiene hypothesis. So one of the things that's observed is that that in very sterile environments where immune systems aren't exposed very often to pathogens, this happens in mice, and maybe it happens in developed countries too, and there's a higher rate of allergies and autoimmune diseases, and so we've developed model studies based on sort of homeostatic mechanisms in the immune system to try to look at these trade-offs between regular exposure and then protection to these autoreactive diseases. And then finally, the thing that we've done most recently in this immune system model has to do with a kind of coevolutionary dynamics. So in here, we're not just going to look at how the immune system itself adapts, but I'm going to have a mutating chronic infection or mutating pathogen on the same time scale as the immune system itself is generating these adaptive mechanisms. And so this is very much like a predator-prey system or an asymmetric combat system where you have a spreading disease and you're coming to try to combat it with your adaptive immune system, and there's a variety of different kinds of things that can take place. So today, because of the appearance of what I would think of as a tipping point, I'm going to focus on the immunosenescence, long-term development of fragilities, as well as this runaway kind of chronic infection that can lead to immune escape or pathogen escape and immune collapse. Okay, so here's the beasts that are in the model, and there's cartoons for them that were drawn by Sean. And the first one is the antigen. So this little picture here just represents whatever your infection is. We will have naive cells. These are generated in the bone marrow, and they emerge with a variety of different receptors attached to them. And it's really the match between a receptor and the antigen that determines how effective the cell is at ridding the body of this pathogen. Okay, the naive cells turn over at a rapid rate in the body, and that turnover is just sort of generated again by the bone marrow producing white blood cells. But when you have an infection, it will create a memory cell. So the response of a naive cell to the infection creates two types of cells. One is memory, one is an effector cell. The idea of the memory cells is the thing, like for instance, when you get an immunization, you create this memory cell which stays in the body much longer than a naive cell. This is sort of days and this is years. The effector cell is the other product of these naive cells when they're presented with antigen. And these are the things that basically go and destroy the antigen, rid your body of it. And the dendritic cells play a role in presenting the antigen to the naive cells and the memory cells in the first place. Okay. All right. So basically, here are the mechanisms that I just told you about. Antigen will grow. You put the infection in your body, it will split and it will divide, and that will happen at a certain rate. The antigen is captured by a dendritic cell. And taken, then now it's busy, and it will be taken to a naive cell or a memory cell, at which point that cell will divide and create these effector cells that can remove the antigen and also remember the exposure that you had. The antigen is removed by the effector cell, and then the effector cell dies. Okay, so all of this is naturally written in terms of basically a set of differential equations. I'll get to those in a moment. But the effectiveness, as I mentioned earlier, of a particular naive cell to rid the body of an infection in the adaptive immune system depends on a match or an affinity between the white blood cell and the antigen. And so here it's represented by a little sort of latch and key response. And this can depend on a lot of different features, connections between these sort of proteins, shape, charge, size, hydrophobicity. The better the match, the closer, the more effectively these things bind. And we are going to borrow a model that was developed by Oster and Carlson in the late 70s, which just models all these different types of chemistry and shape effects and by a shape space. So some sort of a space that represents in an abstract way the affinity match between the pathogen and the white blood cell. And so the closer you are, the better the match. The further apart you are, the worse the match. And the dynamics will depend on this binding affinity, again, which is a function of the position both of the lymphocyte, which is Y, and the antigen in the shape space. So this is a cartoon that, again, represents a much higher amount of biology at a smaller scale, where you're really thinking about how these things match up with each other, something you might want to manipulate if you're actually working on therapies. But this, for our purposes, at the systems level, captures what we're looking for. So again, the equations then that describe all these processes that I told you about on the earlier slide are described here. I have my pathogen is dividing, it's removed, it's captured, then it's removed. Dendritic cells get busy, and so they get a star on them, stimulate the memory and effector cells, and so on. Again, a set of coupled differential equations. Okay, so the other thing we need to do is we need a way to characterize the severity of an infection. And so I'm going to do that talking about a quantity which I'll call the loss. And so the idea is to just describe the loss or severity as being the amount of antigen that is associated with the infection. So for instance, if I'm exposed to a particular pathogen the first time, then over time, those equations, when integrated, will give the pathogen begins to grow, my immune response gets started, the number of effector cells increases, and then and in doing so, it starts the decline of the pathogen. And then eventually the effector cells go away. And again, I haven't shown here the memory cells, but the memory cells would continue. I think they're actually here. They're just so super duper light, sorry. They're so light here that they're this little purple. That's actually the memory cells that are now there and persistent. So I can see them, but I don't think you can see them. Okay, so the idea is then if I'm reinfected with the same infection, it goes away much more quickly because of the fact that I've already got these memory cells in place that will respond to it. Okay, so what does that do from infection to infection in my immune system shape space? If I start off with a completely random initial population of naive cells, such as shown here, and have one infection somewhere in the middle, then after that first immune response, I'll have a heightened population of memory cells in that area. So what would have been a distribution of losses associated with an infection at all these different possible places in the immune system, well, some places I had a gap, so yeah, I'd have a bigger immune response here. It develops a structure so that on the second infection, I basically I'm well protected in this region that's a close match to the previous. And again, this is why we have things like immunizations, because they protect regions in our immune system memory space. We've developed a stronger response, robustness to an infection that we've seen before at the expense of greater fragility, higher losses in regions that are further away. Okay, so what does that do over time? So the first thing that we looked at with this model is this notion of how does this, starting with a random population of naive cells in the shape space, what happens over time when I have through many different infections? So this is showing you over time, through the course of 400 infections, how does this immune system space evolve? So here's, at the beginning, it's random. My losses, infection sizes are sort of, again, fairly uniform across the different types of pathogens I might be exposed to. And then as I go along, if I have infections are being drawn from a distribution that is more likely to see the infections down here, less likely to see the infections at the top. So over time, I have some stochastic process which draws infections from this set of 36 possibilities, and I develop more and more of a robust response to the common infections at the expense of a very large response to the rare infections or infections I haven't seen in the course of my lifetime. So if you compute a particular realization, you'll have some stochastic process starting off at kind of some average value. You can see a trend to smaller infections. That's because you're more likely to get the common ones. But occasionally a spike. Those are the rare infections. If I average over the losses, I'm becoming more and more fit. Here, J is the average size of the loss, so more and more fit, but there's a collapse that happens at the end, right? So I'm getting better and better and better. Here you can see your immune response evolving somewhere here at the beginning, somewhere here in the middle where you're at high fitness. But then when you get to the end, you've got gaping holes in your cell types. So again, these are all the naive cells, the memory cells, and there's nothing here so that if I get that infection, it could wipe me out. Very large loss. So this idea of the natural mechanisms of the immune system starting off with a blank slate where anything is possible, naturally adapting to protect me to the common diseases, eventually that same mechanism leads to extreme fragility of the system. Okay, so if I look at this in terms of the distribution of sizes of the infections or losses in the infections that you see over time, what happens in the original and naive state is I have a uniform distribution of infection sizes. So here I'm plotting the number of infections of a given loss size as a function of the size. I have really small ones, so these are the ones that don't result in very, they're small. It's not a very costly infection. Out here, I have a very large loss, right? And you can see that I start off at the beginning in the naive state. There's, all infections basically produce the same, on average, the same size loss. That's when my immune system shape space population of white blood cells is uniform across the possible infections. A little later on, I will see something like this, where I'm starting to adapt more immune cells to the common infections at the expense of having memory cells that bind, or naive and memory cells that bind to the less common infections. And so I see a broadening of the distribution until, and then later, actually, I get something that looks more like a power law. So the longer I go, again, it's log-log plot, straight line. What that says is the probability of an infection of a given size goes like 1 over size to the power. And so there are some, most of your infections are small, but there's a few infections that are much, much larger. And in fact, when you get out here to the point of immune collapse, you've gone even beyond this, so that you have not just a power law, but you have excess large events, and you can expect something bad to happen. Okay. All right, so this fits into a much broader context of constrained optimization problems, whether the optimization method is one based on fitness and evolution, or whether it's something that's deliberately designed into the system. There's a wide variety of different kinds of systems that we've looked at that produce this sort of, that you look at basically an uncertain set of events, resources that can be suppress the loss, fire breaks or firefighters, in this case of wildfires, immune repertoire, and then there's an optimization or a biological process that's organizing the system to minimize the loss. In fires, we can think of it as fire breaks or firefighters or ecological plant types and ecological sorting. In the shape space model for the immune system, the population of the naive cells are a little bit more like a sprinkler system, in the sense that they have the strongest effect for the antigen types that are close to them and a weakening effect further out. But all of these kinds of problems fall into a similar context of trade-offs between robustness to common events and fragilities to events that are rare. This power law statistics is something that's widely observed in all kinds of phenomena. So for instance, again, this is the rank or the cumulative frequency is a function of some measure of the size of the event. And power laws, it turns out forest fires are a two-dimensional type system. Web files and the internet is a one-dimensional type system. Data compression, the original Shannon coding, is a zero-dimensional version of this optimization problem, which produces not power laws, but an exponential distribution. Let's go do this. And the idea, more important than power laws, is that you have this broad distribution of events, where most files, fires, and infections are small, but the most packets, burnt trees, and most sickness, the greatest loss associated with infection, are associated with the few relatively large events. So high variability is general. It's something we should expect in our system. It's more important than power laws. It's not necessarily a bad thing. It can be optimal, right? It can optimize a cost function. It can be better to have high variability in your immune response. But in the particular case of this failure mechanism, it's gone one step too far and created these excess losses. It can happen as a result of natural mechanisms within your system. Okay. So the thing I want to talk about last is this notion of coevolution. So the idea is now, I've just talked about so far fixed types of infections and what the immune response is. What happens in something like HIV or SIV, where you've got a long-lived infection that itself has a high mutation rate? And so what we did is we took, this is Kimberly's work, modeled the adaptive immune response and added to the pathogen a mutation rate of its own. So we have the same kind of lock and key response between the lymphocytes and the pathogens, use the same kind of shape space model, but in this particular case, the pathogens themselves will also mutate. And again, a close match corresponds to close proximity in shape space, and a bad match means things are well separated. Okay. So let's look at what the dynamics are in shape for an infection. So the equations now, the change in the equations, I'm just going to focus on the pathogen and the total number of lymphocytes because we're always talking about just one infection, so we're not looking at this long-term evolution of memory in this case. And we'll start the system off with lymphocytes at a certain level. This is just the total across the whole shape space, and a pathogen sitting low level of pathogen. As the system starts to evolve, the first thing that happens is you'll see that the pathogen begins to grow in the shape space, and there's some kind of growth mechanism and a sort of a constraint sort of built into it, some kind of a resource limitation to how much it can grow. Okay, then the next thing that you see turning on over here is that because of the pathogen, it stimulated the growth of the lymphocytes, which are now trying to come and catch it. Again, I'm not showing you shape space. I'm just showing you total population. We'll get to shape space in a moment. So it's growing, and as it grows, it's going to rid the body of the pathogen. So the pathogen's turning over here, and then pathogen is coming down. And then after that's true, the effector T cells are rid from the body, and you go back to whatever your homeostatic equilibrium is for the T cells. So here's a limit, basically a cap on how large the overall T cell population can be. Either you can think of that as coming from the biochemistry or space in the lymph nodes, but there's some cap that has kind of a soft top, and the softness of that top is determined by this homeostatic parameter psi. Okay, so that part was just showing you basically what happens if the pathogen doesn't evolve. So if it doesn't evolve, here it is somewhere in shape space. I'm going to turn on my immune response. It's going to be stronger right near where the match is. It's going to be weaker far away, and it will clear the infection. But if the pathogen now mutates, it's running around in shape space. It has the possibility of jumping away from where it was to begin with, and so the population of lymphocytes is chasing after it. It's a predator-prey-like reaction that gives rise to this sort of arms race, if you like. Okay, so let me think about this. So the other thing that we can do is we can plot this as a sort of a phase plot. How am I doing in terms of time? Okay, so maybe I will save this for anybody that has questions about it later. But we can also talk about the different situations in terms of what happens. You have mutation, you don't have mutation, in a sort of dynamic phased way. It's the most important thing is sort of getting to what are the range of possibilities in this system. So in some parameter regimes, depending on the pathogen mutation rate and the width of the binding affinity curve, I'll have early clearance. Pathogen comes in, immune system clears it, everything's done, right? In other cases, I'm mutating so fast that I have early escape. So it's too fast. The immune system simply can't keep up with the pathogen, and it basically runs my immune system out, and the infection, I've basically died. And the intermediate case is one of chronic infection. So in this particular case, what I have is I have the pathogen and comes in, the lymphocyte is working to clear it, and they coexist for long periods of time. And it's within this chronic infection state that we see the richest kind of dynamics. So we've separated this out into a phase diagram. One of them is shown here, corresponds to the case of low homeostatic pressure. And turns out in this particular case, you get oscillations. So you get a rise in pathogen, then the lymphocyte comes, and they sort of oscillate with each other, and that ultimately produces an intermediate band here. In the case of high homeostatic pressure, you have a sort of slightly simpler looking structure in the phase diagram. You can think of this as being a case where the homeostatic pressure is such that it just won't let your immune system build that big, and it's just going to kind of keep it in a steadier, kind of damped looking phase. But in each case, we have three situations for what can happen. One is this early escape associated with rapid mutation. One is early clearance associated with high binding affinity. And then there's this intermediate case associated with chronic infection. And the dynamics of chronic infection is extremely rich. So here's a picture in the shape space of what's actually happening during that situation. I have the pathogen on the top, lymphocytes on the bottom, and you can see here starts the pathogen off, and it's sort of, this is the early phase, and these mutation jumps are moving along, right? Here is the lymphocytes, and they're starting off near where the pathogen is. They're growing, 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 and they're together, but then there's a split. Okay, a split. So what's happening there, right? There's a split in the pathogen, 2 branches. Okay, 2 separate pathogens basically evolving, or different species running away, right? Now what happens in the immune system? For a while, they're kind of close together. The same immune strong response is able to fight them both. But as they get further away from one another, the T cell population must also split. So it's splitting because these have grown out of reach. And then there's another split, and there's another split, and there's another split. And eventually, what happens is you've exhausted, oops, I guess you can't see that here. You exhaust the immune system with a kind of pathogen escape, immune escape, and the system crashes. Okay, so this is just showing how the dynamics of the pathogen in more detail drives the splitting evolutionary dynamics of the T cells. And basically the point is that this coevolutionary dynamics leads to a fragility and sudden pathogen escape in the system. And it's coming about because the same number of lymphocytes has to somehow fight and control populations that are getting more and more diverse within the system. Okay, so I will end with this. So these are just 2 examples of studies that we are looking at in terms of how the very basic mechanisms in the immune system lead to system robustness. Obviously, they help us get well when we're sick, but those same mechanisms can also lead to fragility and breakdown. The immune system is an interesting system to study. So just like the ecosystems of bacteria in our gut and things like that, the body presents a really interesting, contained system to study of mechanisms for complexity that may occur in a lot of other situations. In our particular case, studying these trade-offs have led us, to find a variety of different fragilities of the immune system that arise from the basic mechanisms of driving the system, of which makes the system robust in the first place. To me, I'm very interested in how the behavior of the immune system relates to some of the other systems that we're studying in my group. So one is just the notion that specialization may lead to fragility in other networks. So we've looked at this in disturbance ecology and sort of evolutionary dynamics. I think it's also relevant to studies of learning, memory and learning, and I'll put that as a comment for later. Coevolution and these asymmetric combat scenarios with resource constraints show up in predator-prey systems. They show up in disaster response. There are similar kinds of effects that show up in information diffusion. And one of the observations here is that you can have this sort of splitting into multiple threat zones. So that same type of thing totally shows up in wildfires. We've had a variety of times in California where the conditions are right and we have 10, 14, 100 different wildfires all happening at the same time, and you have to figure out where to distribute your resources for wildfires. And so you need to think about strength, and you need to think about how the system is growing in response to the external conditions and the resources you have to allocate to it. I think I'm really interested in terms of thinking about how to add these system level basically ideas to develop new therapeutics. And so on the one hand, there is the importance of molecular mechanisms. So really understanding at the protein level, what are those bindings, right? And how can we do early detection? How can we catch this early? How can we know out of the wealth of data what's happening, where it's happening, and how to go in and handle it? I think the systems level approaches and network level and dynamics approaches need to be coupled with those microscopic approaches, because you may think you're doing one thing and it has these long-term consequences later. Or you may think that there may be timing issues associated with response that are important. And so one of the things that we're thinking about in terms of timing is how different types of strategies for response can be combined in an optimal way. Let's see, is this here? OK, thank you. I'm done.